Alright, good afternoon students again. This week we will be discussing soil pH for the lab part. I have Sri here with me as well. We have discussed this in the lecture class as well, but I'll briefly go about soil pH and how do we measure that in the lab. Soil pH is the concentration of hydrogen ion in soils, or we can define that in a variety of different ways. Like measurement of acidity or alkalinity in soils. More technically, if you want to define that, is the negative log of the hydrogen ion concentration in the soil. So we are measuring how much of the hydrogen ion concentration is present in the soils, and then based on that we are interpreting the soil pH. If we talk about the soil pH, it ranges from 0 to 14 all the way. 7 is the neutral, it's kind of more acidity. If you go on this one side, it's a high pH but low acidity. Right? So for instance, if you are going below that 7 or neutral, then you have more of a low pH, high acidity, which also means like high hydrogen ion concentration. So high hydrogen ion concentration, high acidity, but low pH. If you go on that side, then typically is low acidity, low hydrogen ion concentration, but high pH. We're going towards the alkalinity. We're going from that direction. We're going this direction, and we're going towards the acidity or acidic soils. Okay? Each unit change in the pH increases the concentration by tenfold. What that means is, if you have a soil pH of 6, if it goes down to 5, then it change in the pH that increase the concentration by tenfold. That means it increases the concentration of hydrogen ion in that case by tenfolds. If it goes from 6 to 5, 5 to 4, 7 to 6, and so on. If it goes that direction, it's kind of like similar concept again. Each unit changes in pH, increase the concentration by tenfolds. So if you are going that direction, it's decreasing. If you're going this direction, it's increasing by tenfolds. So that's a little bit on concept of the soil pH. There are many sources of soil acidity that we typically see in the soils. Cover a few of them here, and we have discussed that in lecture as well. So if we add anything that result in hydrogen ion in the soils, will be the source of acidity. So whatever you are doing, whatever fertilizers you are applying, or whatever activities you are doing in the soils, that instead of adding more hydrogen ion in the soils is the sources of acidity. So hydrogen ion in the solution, or CEC, in the soil system, we do have hydrogen ions already present there. It might be in the soil solution, or it might be in the cation exchange sites. That is the measurement of the acidity. So that's the source of acidity in the soils. In nature, even in natural conditions, we do have some of those hydrogen ions present in that's how it come out. Aluminum in the solution, or CEC. So aluminum is the other one, mainly the CEC, that is also a source of acidity. So if you have any aluminum or acidic cations like aluminum and iron, kind of does the similar thing. So if you have aluminum or iron in the soils, then we typically see more acidity on the soils. So on top of that, some other sources of acidity are functional groups. Organic matter, we do have a lot of organic matter in the soil. So if you have organic matter, definitely there will be some phenolic groups and carboxylic groups present on it, and then those kind of acts as a source of acidity as well. The other one is fertilizers. We apply different fertilizers, and especially nitrogen fertilizers. During the nitrification of ammonium type of fertilizers, you can convert it into NO3, and then it also add some acidity on the soils. On top of that, sometimes we get some acidic type of rainfall, so we get some acidity in the soils as well, and also some plants during the respiration, especially on the root zone areas where there might be some acidity creation happening in the soil underneath of the soil system. So there are many other different types of sources, but these are some of the important ones. Types of soil acidity. We do have two different types of soil acidity. One is the active acidity. The second one is the reserve acidity or potential acidity. Okay? So active acidity is the smallest pool of soil acidity, and it is basically we are measuring the presence of hydrogen ion in the solution. That's what we'll be doing here today. So we are measuring the active acidity on the soils that we collected at the beginning of the semester. Okay, so that's what we will be doing on. The next one is the reserve acidity. Reserve acidity is a bigger pool of acidity in the soils, and typically the reserve acidity is the one that is associated with aluminum hydrolysis. That is what the potential acidity or reserve acidity is. If you are doing liming, then you have to account both the active acidity and potential acidity, or total acidity. That's what we call it. So we are measuring the active acidity today. What we are doing is we are measuring the hydrogen ions present in the soil solution. So we are not measuring the potentiality of acidity addition because of the aluminum or iron type of acidic cations present in the soil. So we are not measuring that, but we are only measuring the hydrogen ions in the soil solution. So that's what we'll be doing on in our lab today. For the potential acidity, there are some other procedures that we can use to measure it. We are not doing that in our lab, but there are some buffer solution, like buffer chemicals that you can use on. It's the same principles, but on top of what we are doing here today, you will add buffer chemicals on that. Again, importance of pH, you all are familiar with that, right? So typically we want to have a neutral pH so that all the nutrients are available for plants uptake, and pH impact the availability of nutrients in many different ways. So for example, if we have pH above 7, some of the micronutrients like the zinc, iron, manganese will be insoluble and will not be available for plant. Even though with the active acidity we would not be able to do the liming calculations, but it will give us some pretty good idea like what is the fertility status of your soil, how your soil can supply some of those nutrients and so on. So this is a picture down here kind of explaining the similar thing. For the lab activities today, for this week lab, we'll be determining the active acidity as I mentioned earlier, right? So this method is commonly used in commercial soil testing labs as well, and it involves the use of a glass electrode, which is paired with reference electrode and then plugged into a good commercial pH meter. That's what we'll be using today as well. That need to be standardized. We'll do 3-point standardization with pH 4, pH 7 and pH 10. The pH of the soil suspended in water is indicated by voltage generated between the two electrodes, and then we can measure the change in voltage in the proportion to the logarithm of the changes in hydrogen ion concentration in that soil suspension or solution. Fundamentally, that's how we measure the pH. Some of the materials required, so definitely we need a pH meter. We do have a graduated cylinder to measure the volume of water that we will be adding on. We do have a balance to measure the soils. We do have stirring rods, and we do have beakers instead of plastic cups. So that's what we'll be doing now. We'll weigh out 10 grams of soil and put into a sample cup or beaker. We'll have a beaker here today, and then we'll add 10 milliliter of DI water. So we are making like 1:1 ratio of soil and water, and then we'll stir that vigorously for 15 seconds, and then let it stand for around 45 minutes. The idea behind that is, in that 45 minutes, the soil will equilibrate, and all of those hydrogen ion concentration in soil would be in the soil solution, and we'll be able to measure that, right? So you can do on 2 reps as well and get the average, but we'll be doing only 1 rep today. And then after we are done with that, we'll standardize the pH meter, and then we will provide the data from your soils that you collected at the beginning of the semester, and then you write a lab report on that, and that lab report is due next week.